name is Kathryn Braden. I'm excited to share my research with you today where we're looking at the role of TLR7 in Oxysterol-GPR183 induced hypersensitivity. This is a follow-up on a recent paper we published showing that GPR183 is a potential therapeutic target for neuropathic pain. In this paper, we found that nerve injury leads to an upregulation of GPR183 in the spinal cord, leading to pain. We also found that direct activation of GPR183 using its oxysterol ligand induced pain in naive animals. And both of these effects were blocked using SA14, a novel GPR183 antagonist that we developed. So this indicates that GPR183 signaling in the spinal cord is contributing to pain states, but how is unknown. So to investigate these mechanisms, we repeated our previous experiment using SA14 given intrathecally to mice or its vehicle at 30 minutes prior to the oxysterol or its vehicle. And as you can see, the oxysterol produces this hypersensitivity or decrease in the polyphenol threshold, which SA14 is able to block. To look at the mechanism for how this is happening, we took the dorsal-hom spinal cord at 2 hours post-injection and ran RNA sequencing to determine what pathways were differentially regulated by GPR183 activation or inhibition. And as you can see in this heat map, there were a number of genes that were significantly upregulated in the oxysterol group in this red color compared to vehicle in this blue color. And SA14 was able to ameliorate a number of these genes being upregulated. We look at the gene ontology of what pathways were associated with these genes. We see a lot of toll-like receptor signaling and innate immune responses, which makes sense when we consider that GPR183's primary known function is in the immune system. When we look a little bit closer at these genes, we see that 206 were significantly upregulated by the oxysterol, 84 were significantly downregulated by the SA14, and 14 genes overlapped between these groups, indicating that they're differentially regulated by GPR183. Of these, there was only one toll-like receptor, TLR7, and several other toll-like receptor-related signaling mediators. TLR7 is an endosomal toll-like receptor, and there's some previous literature to indicate that it may have a link with GPR183. In dendritic cells, it was found that GPR183 was a negative regulator of the type 1 interferon production that's downstream of TLR7 activation. They also showed that TLR7 activation was able to increase GPR183 mRNA. This previous literature supports our RNA-seq data and gives us a better understanding of what may be happening in the spinal cord. But of course, upregulation of a gene doesn't mean it's functionally involved. So to determine whether TLR7 can be functionally important to GPR183's ability to induce pain, we used a TLR7-specific inhibitory oligonucleotide, IR661, to see if TLR7 inhibition is sufficient to block OHC-induced allodynia. Indeed, our preliminary results show that TLR7 inhibition is able to dose-dependently delay oxysterol-induced allodynia, suggesting that TLR7 is functionally linked to GPR183 signaling in the context of pain. It is possible, since IR661 is unable to completely block oxysterol-induced pain, that other compensatory mechanisms are involved, and those will be investigated in future studies. With that, I'd like to thank my mentor, our lab members, our collaborators, our funding, and you for your time. Thank you.